So let's talk about these modification of starches. As I earlier said, it is quite a complex phenomena to understand the biochemical processes uh, for the synthesis of starch as well as their applications. And understanding this is very complex. But as I've given you a summary in the previous table about different known enzymes and their effect on the starch, uh, a little is known, and still there are, as I earlier said, there are limitless opportunities to explore these areas. So in the coming slide, we will see that how these different enzymes will shed some more light on these different enzymes and their role in producing different types of modified starches. Although this is not very comprehensive, these slides have been taken from your one of the textbook that has got more detail. It was not within the scope actually of this smaller presentation to cover everything here. So I'll summarize these things here, and you can refer to your textbook for further details on the same areas. So I've only highlighted some of the enzymes which are known and their effect on the starch synthesis. So if we only talk about this granule bound starch synthase, this is one of the enzyme as earlier discussed. It is involved for the amylose content that are produced within the starches. So amylose are, as you know, that they are the straight chain branches of glucose within this starch structure. So if we talk about this maize variety, especially that has got very low amylose content, that's why they are their phenotypic is more waxy in nature. So that's why they are called as waxy maize. So there's a different actually variety of maize which is waxy due to this phenotype, and that phenotype is due to the enzyme, this enzyme GBSSI, that produces lower amylose content in these crops. So if we talk about cereals, we only know about this enzyme that is very much responsible for a major phenotypic change in the starch. But there are other examples in other crops. For example, we know that there are AMF mutants of potato. Similarly, we know there are LAM mutants of pea and GBSSI antisense lines of potato. They have all shown that they can reduce the activity of GBSS, and then this reduction in this GBSS enzyme activity by these mutants that would produce specific amount of amylose in the final product. So that is how these different mutants can affect the expression of this enzyme, and this expression of enzyme in turn would lead to different ratio and proportion of these amylose content in within this starch component of these crops. And as you can see that these are the major crops which have got starch, and that's why we do a lot of biotechnology research, especially for potatoes and peas and other cereals, because we know that these starch would have got major implication in the industry, and they might have major economic impact as well. So there have been a lot of other studies just to prove that these enzymes, for example, these GBSSI enzymes, as they are produced in other plants, so they can be actually added back into these amylose-free potato plants, so that we can get to know that by adding these enzymes back or expressing their activity in these mutants can actually increase the amylose content in these plants. So that would actually show, or in other words, strengthen our hypothesis that this GBSSI is more involved for production of amylose content in these starches. So certain examples are given in your textbook again, so we can see some of them here. 3.5% to 13% amylose. It has been restored in potatoes that were amylose free varieties of potatoes when we transform this GBSSI enzyme from cassava, because we know that the cassava plant is one of the another source of major source of starch. So it has got more GBSSI enzyme, so this can be transformed into mutants of potato which otherwise don't have high amylose content, but once we bring about this biotechnological change into these kind of potatoes, the amylose content can be restored in these plants. Similarly, we also know some of the wheat mutants that do lack this GBSSI enzyme or activity, and that would in turn lead to a lower amount of amylose, because GBSSI, as I said, it produces amylose in your starch chemistry. So if we substitute the genes that produce functional GBSSI enzyme with some of the null alleles, that can reduce the starch content from initial 25.5, that is the original starch content produced by these crops. It can be reduced to 22 to 23%. So that has been shown by different studies. So that actually tells you that we can reduce this activity of enzyme by substituting with some of the null alleles. There have been other studies where people have actually eliminated active enzymes by 2 and 3 loci in wheat endosperm. That has got much lower amylose content, for example, 16 to 21%. So that can actually reduce the amylose content in your final starch that is otherwise 25.5% in those wheat varieties. So we know that if we have a functional GBSS enzyme from a single locus pair, it can reduce the amylose content dramatically in food, any crop. Similarly, 14.1 and 16.1% amylose content has been also created through one of the mutagenesis. This is through ethyl methane sulfonate mutagenesis of seeds. So these have been different reported studies in which people have actually tried to reduce the activity of GBSSI1, or they have used this enzyme transform other plants to increase their amylose contents as well. So there are different studies. There are more details are available in your textbook, particularly with the mentioned chapters. And a few more things that we know about different studies. There are two functional wxae alleles that are known in case of rice. These are known as wxae and wxb, where wxae produces more amount of amylose, whereas wxb has shown to be producing less amount of amylose in the starch component of the rice grain. There have been different studies that show that wxb allele, it has resulted in ineffective production of GBSSI enzymes. So this is related to the suppression of activity of enzymes, especially in Japonica rice, whereas other studies have shown that wxae allele, it produces larger quantity of GBSS enzyme and in turn higher amount of amylose in indica rice. So there have been different studies that shows that how different alleles, they would have different impacts on the production of amylose content in different varieties within the same crop. Similarly, there are other two wild type alleles in case of rice. They have been reported. They are wxae and wxb again. If we talk about this wxb, it has got 10-fold lower GBSS expression than wxae. So we see that they have got especially different impact on the suppression of activity of certain enzymes, and then that would lead to different chemistry of the starches, for example, lower amylose content in this particular case. So we know that this is due to a point mutation within the genetic sequence of normal rice enzyme wxae allele. So these are few studies. So we talked about this GBSS1, or especially amylose content of starch. Another, as I showed you in the summary initially, that the starch synthase activity is also, we showed that where it is well known that what mutant it causes and what are the effects on the final starch contents. So they are known to change the amylopectin content in the amylopectin structure, especially this starch synthase. Unlike GBSSI enzyme, it has got more dominant effect on amylopectin rather than amylose content. And they have very subtle effect or not any profound impact on the starches or the chemistry of starch. It is known that it does not create very major changes in the starch contents or the function or chemistry of the starches. But some of them, for example, this SSA2A and SS3, they are very well known in case of maize, that they do have impact on the starch synthesis in particularly in maize. In the SS2A and SS3, they were the they have shown to have some impact. Whether the in case of if we talk about rice, the only one known is SS1 mutant. That is very much well known, and it is only reported in cereals in rice. In case of maize, we have this called SS2A, as I showed you in the summary initially, that it produces sugary type 2 mutant. So that mutant is more important industrially because it would be producing maize that has got sugar contents, and that sugar contents would then ultimately impact on the eating quality of these maize which are produced for eating quality. So in potatoes, there are different. The studies have shown that antisense inhibition of SS2 and SS3 has resulted very much modified starches, especially the amylopectin contents are very, very different, and they have shown different impact in the potatoes as well. We know that the endosperm of the maize endosperm, especially amylose extended, is one of the variety that has been found 50 years ago. Actually, this amylose extended actually produces higher amylose content. It can have up till 50% higher amylose content in the starches of different crops. They produce different varieties, and this actually produces a different type of, I would say, a third type of starch that is produced by this kind of mutation. So we have also see that amylose and amylopectin starches. This, as I said, that this rather than having high amylose, high amylopectin, this is sometimes referred to as a third type of starch or an intermediate starch with varying degree of amylopectin and amylose that has been produced through biotechnology approaches. There are also some other evidence that comes from wxae intermediate starch that have got no GBSSI activity, and it would be producing no true amylose in the final product. So as I said, I have given you a summary in the previous slides of different enzymes, and some of the studies are mentioned here in very brief detail, like what has been seen in different studies, that the impact of different enzymes and the different kinds of enzymes and different kind of starches and different kind of crops in which this biotechnology approaches has been tested. Of course, this is not an exhaustive list, so you have to, particularly for these examples, go back to your textbooks and see those details. It has to be some more detail that has to be read through those textbooks so that you understand these concepts very clearly.